All right, so thank you very much for inviting me. I'm honoured to be invited, and I've worked with Dorina over many years. So it's really great to see you all there and around the country, I think. What I wanted to do today is, as Dorina said, I've kind of spent my career working on cohort studies in order to try to improve the evidence base relevant to brain aging and therefore aging, really. So I've been based in Cambridge for all that time, since the mid-1980s, when I think probably before many of the people who are attending this conference were born. So I thought that it would be helpful with apologies to those who've heard me speak before, but to provide the range of results that are relevant to understanding brain aging in populations and how they fit with sort of current societal and research efforts and how they can inform those efforts. This picture itself is, of course, Cambridge is known for its lovely architecture, its intellectual and cultural life, but we also have inequalities, and down in the left-hand corner is a local newspaper photograph of our rough sleepers, which of course is more of a current phenomenon. It's sort of something that emerged over recent decades and is a real issue of concern, and people who are homeless are underrepresented in our research. So I'll come back to that kind of issue, I'll touch on it through the talk and then come back to it more at the end. So now, to try and make sure, yeah, okay, has it progressed all right for everybody? Yeah, great, thank you. So I'm going to talk about population, older people, dementia and society. So those are kind of key parts of my title. And that takes me through demographies, global considerations, the Alzheimer's disease narrative in the UK at present, dementia, what it is, syndrome, and evidence from true populations, as I've mentioned, and then a sort of stock take to try to kind of pull it all together. So the demography, you'll be well aware, is really quite stark. This is from our World in Data, which has fantastic data from around the world. And this also gives projections. And these are the projections that it has going up to 2100. They may change a little given the current configurations with COVID and the mortality associated with that and with conflict and so on. But basically, there's going to be an increase in the older old. I hope you can see it on your slides, but at the top is 90. So you can see that basically the pattern is that the increasing proportion of the 60 or the 50 plus population will be in the older old age range. So it is vital that we have research that reflects that older old component as well as the important aspects of mid and early later life. And this would be a dynamic slide, but I thought it was too much of a risk. But also in our World in Data, you can look at the median age change across time, and it splits the population into two equal groups on the basis of estimated population demography. Of course, some countries have better information on this than others for all sorts of reasons. But this is where you split it at 50. And here you can see that the median age of 50 and above in the northern hemisphere is already very strong. So basically, as many people aged over 50 as below 50. But in other countries, there are enormous proportions, as we know from the media every day, enormous proportions of younger people. And it is not only about the current older people that we need to think, but the future older people, because in many parts of the world, people are experiencing very poor early life, which will have longer implications right into their aging if they survive. And this is in the context of the size of the world's population, and this is really, really stark. And this goes along, of course, with climate change and our need to address issues which are of societal importance, not just dementia. So dementia occurs in a context, and it's one of the things that I have been very aware of, given the epidemiology and public health background that I have, is the context of the work in dementia. We can't do our research completely dislocated from all of these things going on in society. And of course, that leads me on to the Sustainable Development Goals, in which if we were able to do these things across the world, we would have healthy aging. And I'll come back to that later. So completely different and perhaps dislocated. Sometimes I've characterized this as lots of hot air balloons. Not necessarily that there's hot air, the hot air, it's hot air. I'm not being negative about the research that's within it, but the hot air balloons which are no longer tethered to the ground. But there is an awful lot of research which is going on, which is very, very wonderful in its own right. But sometimes it can seem as though it's become untethered from the reality of dementia in the population. However, the recent decades have seen the dominance of a particular theories about dementia syndrome, and what proportion of dementia is, as it were, caused by particular types of pathology. And you will discuss quite a lot of this in the conference in the next couple of days. People have dedicated careers to these things, and they are very important. So here we have tau and microtubules and phosphorylated tau and so on, and amyloid angiopathy and plaques. And of course, these things are incredibly complicated, and they occur within a very complicated organ. So we really need to understand, as Andrew said, the kind of normal aging, usual aging, if you like, and pathological aging, and what is really going on and how do systems interact. And again, I'll talk about these. A lot of these things are examined rather in isolation, and our role as, I think, in cohort studies and in much of systems biology is to try to integrate and think about how these things all fit together. And I'll come on to that a bit as we go on. The current environment is very fevered. So these are just a few things from the internet in the last few days. And we have the promise of being fed to the public about genes and Alzheimer's being incredibly important, also about therapies and biomarkers, and the trials being launched at the moment, the therapies themselves for Alzheimer's disease that are currently under scrutiny by the MHRA and NICE. And then, of course, the individual choices we make about, and the diets that we're being fed from the media about particular risk factors, from blueberries to broccoli to Sudokus and so on, and how to make sense of all of that. So I think the public is probably pretty confused. So then we come back, so from public health point of view, we say, well, what is it that's of concern to society? Well, it's the dementia syndrome. It's not actually the subtypes, it's the syndrome itself that has these major impacts. And of course, needs to be understood much better. And I spent my career trying to do that. And there's still so many unanswered questions. But the syndrome itself is dimensional, as you'll all be very aware, influencing cognition, sufficient to interfere with daily functioning, and with exclusion criteria with particular conditions that may cause apparent dementia syndrome, but aren't, like delirium. And these are all critical for the diagnosis of dementia syndrome, and each of those is not categorical. So it's not you have it or you don't, it's all contextual. Cognition is in context of many things to do with the way your life course and your society, as are daily functioning. Daily functioning changes. We have functioning, which is kind of key to our being able to eat and clean ourselves and dress ourselves and those sorts of things. But also then there's the kind of more complicated things of being part of daily life. And of course that changes across time and is very different across cultures. So these things are not easy to kind of pin down necessarily with a binary, but we have to do it in order to create a diagnosis. And then we have the subtypes and possible causes. Well, age is very, very important and still dominates the risk profile. And then we have, if we look at the subtypes of dementia, what I'm saying here is that age is associated, possibly dependent dementia. So I'll come back to that in a minute. But then if you look at the subtypes that we have characterized at the moment, you can see there's a whole raft of them, but there are many, many more. And there are things that affect cognition that may not cause dementia. So there are lots of considerations when you want to study dementia syndrome in populations, and when you want to think about how to address those individually and collectively. And not least the whole systems of the body and the way that our bodies interact. We have perhaps for some years seen the brain and the nervous system as being separate from the body and the other systems, but increasingly over the last few years, we've begun to reintegrate, I think, the systems of the body and understand and look at and examine what the impact of things going on in the body might be. And Andrew mentioned inflammation, and that's been knocking around as a kind of a possible etiological concern in dementia for some time. So I started out by saying, well, I need to understand what dementia is in the population, how the variables associated with the diagnosis of dementia syndrome are distributed in populations. And so that's how I started out in the mid-80s. So that's now 40 years ago. And I became involved in, well, I did on my own, the study in East Cambridgeshire with support of Dr. Calloway. And then in Cambridge City Over 75 cohort, I worked with Felicia Huppert and Jean Paykel. And these are all names from the past for people, but Martin Roth was around in Cambridge at the time that I moved into Cambridge. And the cognitive function and aging studies thing that Dorina mentioned then came on later in the 90s. These are all population-based, truly recruited from populations using general practice registers, which are population-based in geographical localities. So these are these pictures represent the three studies. One is the Fens, very flat and agricultural. The middle one is Cambridge City, a different part, the one that you don't see normally in one of the rather more deprived areas of Cambridge City, but the Cambridge City cohort recruited from across the whole of the city, and then the cognitive function and aging studies. So I'm going to skate through some of the results from these studies to give you a sense of the evolution of understanding about populations and dementia syndrome in populations. So the Cambridgeshire study, the Fenlands in the Fens, was in a defined geographical area near Cambridge. And I interviewed almost all women from a GP practice who were aged 70 to 79 in 1985 using deep phenotyping, as it would now be called, and basically using state-of-the-art at the time, which was very much cruder than what we have available now. But I basically collected on all these women, which was around 90% of those people aged 70 to 79. I basically collected the data on them that you use to make the dementia syndrome diagnosis. And when you look at the distribution of the variables, they're all continuously distributed if you have enough if your variable has enough kind of items, as it were, because most of them are not binary. Or you have a collective of items, such as the mini-mental state, where some might be binary, but they add up. I'll come on to that in a sec. So I knew that I wanted more biological measures. I took blood, and I examined people. I examined them for cardiovascular system and neurological system, most particularly, and in their home settings. 365 women, 2-hour interview usually, and I also interviewed informants for 360 of those women. I tried to follow up for autopsy, but in those days, brain donation was not a normal phenomenon, and that wasn't really possible. But from that very first study, many of the issues that are still around today, like proxies and quality of proxy interviews, informants, and the JORM items came out of that. The instrument that I used included a standardized interview for informants, and that was called Candex. And that really kind of set the base for many other studies that came after. And a lot of the issues are quite difficult, and we still haven't solved them. And maybe it's because they can't be solved. I don't think AI will solve them. So just an example of the distribution of cognitive function. Here we have people aged 70 to 74, the women aged 70 to 74, and the women aged 75 to 79. And you can see absolutely what you would expect, which is the mini mental state distribution. And you'll all be familiar, I'm sure, with the mini mental state, with all its foibles and flaws. But it is in this population, which with relatively low education, you don't get a ceiling effect particularly, but you do have the age effect cross-sectionally, that the people in the higher age groups have a lower distribution of their mini mental state, with a pulling out of the lower scores, with people down here. I think you can probably see my cursor. Can you see my cursor, Dorina? Yeah, so down here being the people who would have had a diagnosis of dementia, but there was some people up here as well. So we're using DSM-3 at that time for the diagnosis of dementia, and it was a clinical diagnosis based on the criteria from the CAMDEX interview. So that was just one snapshot study in the Fens. Then we have Cambridge City over 75 cohort, which is population-based, set up by Daniel O'Connor, Penny Pollitt, Doctors Brooke and Rice, GP and old age psychiatrist, in Cambridge City to look at care patterns. And it was totally population-based, as my study was, and we were running in parallel in Cambridge at the time. I'm setting up these studies. And so both of these studies are truly population representative. And that study wanted to look at the impact of a community resource team with multidisciplinary folk in it: social workers, occupational therapists, physiotherapists, old age psychiatrists, and geriatricians. Does that sound familiar? It does, doesn't it? And it was looking at rates of institutionalization. It actually found that where you had a multidisciplinary team, more people went into institutions. And it was probably the recognition of need in the population at the time. Unfortunately, political changes came in, and the multidisciplinary team that was very effective and was liked by the population was changed. Funding models changed. There was no longer. It was much more siloed, if you like, and it's taken a long time to get back to that sort of discussion about the need for integrated care for people who might have dementia and supporting them in the population. So we were able to pick that study up and move on to look at changes in cognition and function with aging, and that was with Felicia Huppert and Jean Paykel, as I mentioned. And the study had begun in 1985 with screening for dementia, with over 2,000 participants and repeated surveys. And that had, like my study, had 90 very high response rates, 95% in the case of Cambridge City. And we carried on seeing people for up to over 20 years. And we had a brain donation program which built out of the work that I tried to do in the Fenland study, in the East Cambridgeshire study, and also built on the Cambridge Brain Bank. And the methods used for brain donation were developed by Andrew O'Sullivan at that time and Claude Wishick. Claude was working with long-term institutionalized populations and looking at brains, and he was working on tau. So that's kind of come to the fore more recently, but at the time there was a lot of work between Roth and Wishick on tau. All of the people in this cohort had died now, but we were able to successfully have brain donations from over 250 from this particular cohort. This is just what it looks like when you have a flow diagram of a study. And so you have the baseline here with over 2,000 people taking part. And then at each time you lose people, some people come back in again, and you follow people up for many years. And here we go up to year 28. Where we only have 3. So we followed people up for as long as we possibly could with the funding that we had. And these are very difficult studies to get funding for, but we did it as best we could. What it did was allow us, because if you study dementia, you have to study so much about health, because you can't make a diagnosis unless you understand the person's context. As I mentioned at the beginning, unless you understand their context, you don't understand whether their cognition or their function, their daily function, is appropriate to that context. So you have to look at health and medications and all sorts of things. and quality of life and so on. And because we were doing brain donation, we were able to look at end of life as well. So we were able to look at all these different types of things in this study, not just dementia, which is, it's a real privilege to be able to work with populations like this. And I've not put function here, but basically dementia, cognition, and daily function have all been focuses, and many students and PhDs have been written on these types of things here. And we've contributed to the epidemiological neuropathology work that is led from Sheffield. And I'll say a bit more about that later. So this study, from its first round, as my the East Cambridgeshire study, contributed to a European exercise called Eurodem. And Eurodem was an EU-funded project to bring together those studies which had robust methodology and were estimating prevalence. And this is still a very controversial and current contemporary issue. How do you estimate the number of people that might meet a particular dementia syndrome diagnosis set of criteria? And these were countries where a population-based study had been done in a geographically delimited area, where the methodologies were sufficiently robust and sufficiently aligned to be able to bring them together. And Bert Hoffman, who's now in Harvard but was in Rotterdam at the time, coordinated that. And that led to this, which was the prevalence estimation. So prevalence is the proportion of people who might meet a particular diagnostic set of criteria at any given time. And these are the same data in both of these graphs, but here you've got a logarithmic scale on the y-axis, and here you've got an ordinary scale. And you can see here you've got this very sharp rise. So here I come back to the age-dependent, age-associated. If it's not age-dependent, it's very, very age-associated. The dementia syndrome. And here you can see just how linear that is on a logarithmic scale. So you've got a very sharp increase. And so statements like doubling of prevalence with every 5 years over the age of 65 began to come into current into the language at the time. And this helped inform policies. And of course, it fed into the need for a bigger research program, because at the time that I started out in dementia research, you could really basically you could read everything in the literature, which was related to dementia research, including all the animal studies and all the hypotheses about, which are very similar to the ones now, but they're just much more refined now, about what might be underlying dementia. So what could we do in that Cambridge City over 75 cohort on exploring the continuum? And here you can see one of the early studies that we published, which was about the continuous distribution of change. And this is the change over a 2-year period in the population that we were able to study at 2 years, which was only, it was a good follow-up rate. It was about above 80% of people who had survived. We were able to follow up again. However, I was very aware when we did this research that we were missing people who had dropped out. So there's this group at the bottom here who are more likely to receive a diagnosis of dementia. But in fact, if you look at the people who do receive a diagnosis of dementia, it comes across the whole distribution there up to sort of into this area here. So we were able to work with biostatisticians, and of course you saw the flow diagram. We followed people up over a very prolonged period. This is an analysis from the 10 years of the data. And what you can see here is what happens if you look at different groupings and then you model in different ways. So here you have different age groups, and you can see that the people who took part, so the people who were still here and took part all the way through, started higher and higher, much higher for the 80-84, slightly lower for the 85+. If you look cross-sectionally at each time when you do your cognitive testing, You can see that there is a slight decline here, slight decline, slight decline. But if you put in the people who are dropping out at each stage, you begin to get something else. Missing at random, it's called. You get something else. So people who are dropping out are those who had worse trajectories. If you do it matching the people who've dropped out with the people like them who are in this group, these groups here. And then if you know that the people who are in these groups probably don't represent the people who are dropping out and dying in between the waves very well, then you get this here with reasonable assumptions. And with even, you can kind of extend it even more with a truly informative missing model here. And then you can see that there's a very, very different patterns. And it is really important that we understand that the people we see in research studies are not necessarily representative of all those that we are seeking to serve in the population. And that, although it was published some time ago, those findings remain as relevant today as they were at the time. We were able to look in the brain donation study in an unselected way at the occurrence of different Alzheimer type pathologies and vascular type pathologies. It was very important at the time that we included vascular. In the reframing of diagnostic criteria, Alzheimer's disease sort of separated away from the vascular angle, even though we know that vascular pathology is very important, and that's been known for a very long time. But if you look at these distributions, the light grey are the people who died in the CC75C study without dementia, and the dark grey are those people who died with dementia. And what you can see here is that the Bragg staging, which is sort of more tangle-based, and the steroid staging, which is plaque-based, so classical features in the brain of Alzheimer's, the Alzheimer's diagnosis, are very much present in those people who have died without dementia here. And these are all people in their 80s and 90s, yeah. And the same thing with the vascular pathology, there's a huge overlap, and with vascular mixed disease. So you can see that there is not a binary. It's not all the people with tangles are dying with dementia, similarly for plaques, and similarly for vascular pathologies. It's much more complicated than that. You can reach great old age with these pathologies and not die with dementia. And this is very, very important for how we handle findings based on younger populations and promises of or an interpretation of the importance of particular pathologies. So now I come on to the cognitive function and aging studies. And here we have the sites of the different locations, all geographically defined, rural, urban, north, south. And it found very similar prevalence estimates to those that I've already shown you for CC75C. But we were able, with our longitudinal follow-up, to look at things like incidence of dementia, how much dementia is occurring newly in populations without dementia at outset, in a true population manner, estimate the number of people with dementia in the whole population in the United Kingdom and England and Wales, and also look at things like median age at onset and survival, and what might predict survival. And functional ability was the best predictor. So once you have dementia, things like education are not so relevant. It's more how you are. We were also able in this study to look at dementia at death, which I thought was a very important thing, because that's basically what people fear, is they don't want to reach old age and die with dementia. And so we looked at, in 10,000 deaths, we looked at whether people had dementia before they died. And so this is dementia, the prevalence of dementia at death. And you can see that it rises, or severe cognitive impairment. You can see it rises very dramatically and gets above 50% for men, or not quite 50% for men, but 50% for women. And if you look at the in the different different life course experiences, non-manual, manual here, and education here, you can see that there are differences between these groups. They're relatively small, but quite important and highly significant. So this is a very important finding, because this is one of the things that people are concerned about. How long, if I'm going to have a long life, will I develop dementia during that life, rather than necessarily whether they develop it at any given age. So we were also able to look in an unselected way at the pathologies and the interaction of age with the pathologies at death. So this is again from the neuropathology. So we have atrophy, neurotic plaques, tangles in different parts of the brain here, the hippocampus and the neocortex, and the age at which people died, and whether they died with dementia or without dementia. And here can you see for atrophy, it seems to be that at whatever age that you are, atrophy is a bad thing, and it maintains its relationship with dementia. But for plaques and tangles, the picture is not the same, because what you're seeing is that it's sort of coming down or level in those people with dementia, in terms of the proportion of people dying with those pathologies in their brain with dementia. But those people without dementia, you see it's coming up here to meet the prevalence of those pathologies in the brain between the groups, so that the people without dementia are beginning to look, their brains are beginning to look more like people with dementia. If you gave it blind to a neuropathologist, they might say, well, this person would have had dementia, but actually they didn't. And this is, again, really important, and it confirms those findings earlier from CC75C. And this was a big enough sample to be able to examine this kind of thing from a biostatistical point of view. We really do need quite large samples to do this. And these are findings that have been replicated in other neuropathological studies in other parts of the world, and I'll come on to that later in a moment. So what else might be going on in the brain, and might more generally, in that sort of more thinking about things from a more contextual point of view? So we were able to look at the whole concept of cognitive reserve and brain reserve, which is very important, and I think it's a growing field, although it's gone a bit quieter recently because of the dominance of the Alzheimer's research outputs and trials and so on. But the question is, well, what's going on in the brain that can protect people from dementia? So here, this is just an observational slide, which is influences on the expression of dementia of education. So for this analysis, we brought together 3 population-based studies, one from Finland, the Cambridge City over 75 cohort, and CFAS. So we have one which is 65 plus, one which is 75 plus, and one which is 85 plus. So Vanta is 85 plus. And in this study, you can see that the expression of dementia before death is at the group level, is influenced by the level of education that an individual has received. So it looks like individuals can compensate for pathology in the brain through other brain mechanisms. And it's consistent across the levels of, in this case, cortical tangles. So this is quite, this and a lot of other research that's provided further evidence. Of course, it's not all in there, but it's a body of evidence, suggests that there is a very strong cognitive reserve or brain reserve capacity in the brain to protect it from dementia expression during life. And that's just coming back to that, it's the dementia syndrome that's important during life, not pathologies, in terms of, if we're thinking about what's important to individuals and their families. So how do we build from these studies? Those are all quite old, those papers. Much more recently, we were able to bring these studies together. So these 2 of these studies, not the Vanta study, but the Cambridge City over 75 cohort, the CFAS study, and then some American studies which have been conducted, and bring them together, harmonize the data, which is no mean feat, and look at the neuropathologies that were expressed in the brains across those populations. So basically made them comparable, and then combined them in a very careful analysis, really carefully done, looking at 6 key neuropathologies that are dominant in our thinking at the moment. So we have plaques, tangles, and then infarcts, Lewy bodies, which is the pathology associated more with Parkinson's disease and Lewy body dementias, or and recognized now to be very closely related. And then a new phenomenon, which used to be sort of, it's like hippocampal sclerosis, and these are all associated with different proteins. So we have amyloid and we have tau and beta amyloid, and we have alpha-synuclein, and here we have TDP-43. So in these brains, which number now many thousands because it's a big sample, pooled sample, you can see people with dementia in the blue, and in the orange you have no dementia, and the red, it's missing, because some people you just can't tell because they have severe, say, sensory, or have had a severe stroke, severe sensory impairment, and you just cannot tell whether they had dementia before they died, because you can't really test it in that individual. And you can see that here, this is a way of characterizing the types of pathology that they had in their brain. One here, just one pathology of a particular type, 2 pathologies, 3 pathologies, 4 pathologies, and 5 pathologies. And you can see who died with dementia, who died without dementia, and who was missing in each of these categories. And what you can see is that a fair proportion of people had dementia, even though they only had none or one pathology of different types. But as you go up, you can see that the most of this population of brain donors from population-based studies had mixed pathology. And this is really, really important for the dementia research world, because we can't look at things in isolation. We need people to look at things in isolation, but then we need to put them into that bigger context. And that's what's really important. And we're not terribly good at doing it. But you can see in these age groups, these later age groups, not age groups, these mixtures of pathologies, the proportion of people who have dementia in those populations is much greater. So that if you look at it in as a whole, the majority of people who have dementia in these studies have mixed pathology. And they are population based or community derived. Okay, so that's really important. And a lot of that is vascular. So just coming on to the issue of what's going on in the general population, coming back to that sort of systems and health throughout the life course and general health, you can see here that we have age standardized death rates from cardiovascular disease from 1979 to 2013. And you can see there is a very dramatic drop in the United Kingdom as a whole across those periods. It has actually leveled out a bit now, but that drop is really quite remarkable. And so we in CFAS made the argument successfully to the MRC that we should look at generational change. And we were able to raise the finance to look at 3 sites, Cambridgeshire again, Nottingham again, and Newcastle again. These were the ones with the most divergent prevalence estimates from the 1990 round. So in 2010, we went back out to the populations. We replicated the methods. We resampled the population 65 and over. So there wasn't any relationship between the earlier study in the 1990s and the later studies in 2010. And here you can see the prevalence estimates, which show that in the age groups in which dementia prevalence is picking up, there was a reduction in the prevalence using the same diagnostic methods between the first study and the second study. And it's corrected for differences in response rates. And this is a really, really quite remarkable change, and one which I had not expected. Is that happening across the world? Well, yes, it is. Bert Hoffman again convened a group of the small number of studies that were possible to pool into, well, in which this kind of analysis could be done, where the methods were kept steady across time, across generations, different designs in these studies, but basically there was a pooled analysis. And these studies resulted in this finding, which, of course, given that it was combined with different methodologies, it's even more robust, shows this very marked drop in the incidence. In this case, it's the incidence of dementia across decades. And that's happened without any dedicated or targeted pills. And it's something to do with society and how those countries, those high-income countries, have done things in society and probably across the life course. So here I want to mention the life course approach that the Lancet Commission has effectively developed across its timing, given that there have been now 3 iterations of the Lancet Commission. There's one under review at the moment. And that exercise built on earlier work that had been done in the States and by our group to try to work out how to estimate the proportion of dementia that might be accounted for by any given risk factor. And I know the audience will be very familiar with this, with these papers, but it really has brought to prominence the mixture of risk factors across the life course that are amenable to intervention and that may have changed across time, not just cardiovascular disease, as I in the introduction. But it is very important to note that many of these things, like less education, obesity, smoking, physical inactivity, depression, all of these things are associated with inequality. So I'll come back to that. And the profiles of risk factors are very, very clearly associated with the way that life courses play out. So here's one example, just with stroke. Stroke doubles your risk of dementia. But if you look at the index of multiple deprivation in the country, you can see the incidence rate is very highly associated with deprivation. And the same thing for intracerebral hemorrhage, which you might not have expected, but there it is. Ischemic stroke is perhaps more associated with all of those other risk factors that cluster together. So, and the same thing for the risk factors for stroke, diabetes, atrial fibrillation, and hypertension, also all strongly associated with multiple deprivation. And at the moment, we're experiencing quite a major change in the population. We've got here, that's climate. This one on the bottom is climate change. It's our moorlands on fire. But here we've got the homelessness, as I mentioned, and food banks. And we can see that life expectancy is changing in the UK. So so health across the life course is changing, and we can expect brain health to change alongside that. And that's not captured in these kinds of headlines. And it's much less easy, if you like, to capture the public's attention with these things. But it is really important, and it's very important that our policies address these things. So a few tropes debunked. Okay, diagnosis. So at the moment in the country, we have estimated estimated prevalence, which is taken from the studies that I've described today. So it's taken as a truth, using effectively our study diagnosis applied to the entire population in the UK, sometimes with a bit of nuance about inequalities, but that can actually work against services rather than in their favour. And so the diagnosis that comes through in routine records is then matched against the estimate based on the work that I've presented. And that is, of course, not, it's not it's not the truth, as it were. It's our estimate of what the truth might be. We did those estimates in 2010, so they're quite out of date now, and they really need updating. And it doesn't really tell you about any given population. It tells you, in general, what you might expect. And it's helpful for policy, but it's perhaps not sensible to say to a GP, because your age-sex profile looks like this, you have this number of people with dementia in your population. Because, of course, there are confidence intervals around our estimates. So there's a huge range of the number of people that you might expect to have dementia. So it's kind of strange to use our data in that way, but it has been used in that way for many years now. Another trope is 60% of dementia is caused by AD. I hope I've given you some reason why you might question that. In our attributable risk studies at death, it's 20%. So atrophy, old age, and vascular and mixed pathologies all play their part. So I hope I've given you a food for thought about mixed pathologies, and treatments of a single protein, therefore, even if it's highly effective, may only work for a small proportion of people. And those people would be likely to be in those younger age groups where I showed you that slide of the fact that in the younger age groups, the difference between the brains of people dying with dementia and without are very different in terms of plaques and tangles. And the final trope, which I haven't talked about specifically, but I can see it's shot through the whole presentation, is that when people say so many people, it might vary across the world, develop dementia every day, that is not dementia. Dementia syndrome does not develop like that. It is not like being hit by a bus or having a stroke. It is much more often a continuous process, and even when it's sharper decline, it's not developing it every day. So please, I hope you can call those out when they happen. So where do we put our efforts? Prevention of disease, early detection and treatment, or reducing complications? Well, many years ago, Carol Jagger in Newcastle did a very elegant piece of work, which was looking at if you invest in these different areas, what bang for your buck would you get? And those models suggested that if you went for this, and this was even before we'd shown that the dementia prevalence had declined age for age. And the combination of primary and tertiary is where you get your best evidence. But where are we investing? We're investing here in this middle group with early detection. So we need to be sanguine about our ability to change disease by doing secondary prevention. We need to think and balance up the investment and the research activity across all these pieces, which is many people are very aware of that. And this is important for almost certainly for early onset dementias, those people who are known to be at high risk for families, but it will not address the majority of dementia which occurs in the older old, I should say. And so working on general neurological health, it's quite clear, and the WHO has got a very nice report which looks as though it's focused on epilepsy, but it actually is about brain health. And we've worked on this in Cambridge to try and think about having a model which brings primary, secondary, and tertiary prevention all together, and thinking about the social and commercial determinants of neurological health, including dementia at the end of life and in the older ages, and trying to integrate findings that we know about from basically across public health, across epidemiology, and across the dementia world, in this case, to our brain health in later life. And this is a model perhaps you can see where your own research fits into this and how it contributes to this bigger picture. And I think that integration is very important. So how does this all add up? What's truly preventable? How can we reduce risk across the life course through policies, societal structuring that reduces all our risk at all life stages? What is the small and what is the, well, we can actually estimate this, but what is detectable at an early stage? And do we have enough knowledge about natural history, and how can we design trials that really tell us that a later outcome is averted? And that's the kind of evidence that you need for screening. And we need that. We really, really need that if we are to be implementing anything to do with early detection. And it is likely to be a relatively small fraction for the reasons that I've given. And what can we do to mitigate and support by appropriate interventions quality of life for those people with dementia and severe cognitive impairment? Non-pharma and pharma, ideally combined, if we can work out how to get those trials off the ground. And I think we're beginning to see a change in attitudes on that. I very much hope we'll see more trials like that, where we combine approaches. And how can we ensure the best possible end of life for those people living with dementia, with cognitive and cognitive impairment, for the reasons that cognitive impairment is an important category, missed by much effort at the moment. You know, you have to have dementia or you have to have mild cognitive impairment, which is not the same as cognitive impairment, not dementia grouping. And how do we tackle inequalities? And how do we do this without costing the planet? And there's just been some work on American Medicare bankrupting the US, a nice piece in the public. We need to work out how to do these things in a way that doesn't bankrupt us or cost the planet. So thank you. That's my talk, and back to the Sustainable Development Goals. And here, I mean, there are so many people who've been involved in these studies from all sorts of universities, and also, of course, many funders, and hundreds of thousands of interviews, and wonderful participants and their families, and all of the systems that have supported this research over the decades. Thank you. Thank you, Dorina.